So recombination frequencies, calculation of recombination frequencies, as we discussed before, allows you to construct genetic maps. Genetic maps actually take advantage of the observation that physical distance between genes on a chromosome relates to the frequency of recombinations. Farther apart two genes are, higher frequency of recombination you observed. Closer they are, recombination frequency are lower. So how do we measure, or how do we define the distance? We define it, as we mentioned before, by map units, which equals to recombination frequency, which equals to centimorgan. You remember, 1 centimorgan equals 1% of recombinations. Now, let's see how this relates to relative mapping. If you consider this example, you have gene A and B, which express, or for which you observe 20% recombination frequencies. Afterwards, you look at another gene, gene C, and you see that recombination frequency between A and C is 8%, and recombination frequency between B and C is 12%. So what does it tell you? That genes A and B are the farther apart, and gene C must be in between of them. So if we convert recombination frequency into centimorgan, you will see that gene A and B are 20 centimorgan apart. Gene A and C are 8 centimorgan apart, and gene C and B are 12 centimorgan apart. So I will stress again, this is just a relative position, because as we will discuss afterwards, there are double crossing over, there is interference and so on, which actually affects our recombination frequencies. So if you want to have exact map of the gene on chromosome, you have to construct physical maps. And physical maps are calculated based on distance along the chromosome, expressed as number of base pairs. So in order to construct physical maps, you will have to sequence the genes. Now, I would like to stress 2 points about constructing genetic maps. Firstly, you recall, if you have genes which are located far apart and they are in 2 different linkage groups, so the genes exhibit 50% recombination frequencies, you cannot really distinguish between these 2 linked genes and between independent assortments. So you cannot really construct genetic map based on those genes. Second point I would like to make is that the frequency of recombination is usually underestimated due to the double crossing over taking place between 2 genes on the same chromosomes. Therefore, when you construct genetic maps, you always have to keep in mind that the position you calculate based on recombination frequency is only relative. So let's look at this example here. Let's consider these homologous chromosomes which undergo crossing over, single crossing over and double crossing over between these 2 non-sister chromatids. So what would happen if this 2 non-sister chromatid undergo single crossing over and double crossing over? I will just outline here these 2 chromatids right here, this one and this one. So those 2 which undergoes crossing over. So one which is red carries dominant allele for both genes, so you recall it is this position, and second one in blue carries both recessive alleles. Now, what would happen if there is one crossing over taking place between genes A and B? So we recall, if this crossing over take place, you will get recombination, and the outcome will be that one chromosome, this chromatid, would carry now dominant A and recessive B, and second one would carry recessive A and dominant B. So after one recombination, the gamete which will be formed will be recombinant gametes. However, if second crossing over takes place here, this was first one, now there is second crossing over, what would happen? So basically the alleles will be returned to the original parental position. And double crossing over, after double crossing over, the gamete which will be produced will be AB and AB. So these gametes will be identical to parental gametes without crossing over. So keep in mind that double crossing over alters the observed frequency of recombinance, and double crossovers are more frequent between genes that are far apart. Therefore, if you construct genetic maps, those which are based on short distance are usually more accurate than those which are based on longer distances. So how do you construct gene maps? You have to conduct a series of test crosses where you test cross individual which is heterozygous for a different pair of genes, and based on the obtained result you calculate recombination frequencies. You can do it as a two-point test cross when you follow two genes, or you can do it at a time, or you can do it as a three-point test cross when you follow three genes at a time, meaning in each cross. Now let's first look at two-point test cross. So let's consider that you are following genes A, B, and gene C, and you're conducting series of test crosses where you test cross individual heterozygous for AB, heterozygous for AC, and heterozygous for BC. And recombination frequencies are outlined right here, and you see something different here, MU. Map units. Map units equals to centimorgan equals to recombination unit. It's just to confuse you a little more. There are different units because remember, this is not exact values. These are not base pairs. This is just arbitrary units, and arbitrary we can assign different units to it, but each of them equals 1 mu, 1 cm, 1 ru equals 1% of recombination. So let's go back to our test cross. So what does it tell you? You see here recombination frequency. So first what you look at is what is the highest percentage of recombinations, which two genes have the highest percentage of recombination, and you see here it's gene A and C, it's 15%. So what does it tell you? That gene A and C are apart 15 centimorgan or map units. So basically they are one which are on the outer map, on the outer side of the maps. Now, since you know that A and C are outside, so B must be in between of them. And you look on relative distance between A and B and C, and you see that gene B is 5%, 5 map units from A, and it is 10 map unit from C. So basically it has to be, because it's 5 and 10, so it has to be in between of gene A and C, and it's right here. So gene A and B are 5 map units apart, and B and C 10 map units apart. So if you do series of test cross with heterozygous individual for two different genes in two-point test cross, by the series of different test crosses, you can actually define relative position of these 2, 3, 4, or even 6 genes on the chromosome. But you always have to pick up these two genes which are which shows recombination. Let's now solve and answer practical application or an answer two-point test cross with series of crosses between genes A, B, C, and D, and there is something different. In this case, you see you observed crosses where you have 50% recombination frequency. So how do you proceed under this situation? Because you know that 50% recombination means that these two genes are so far apart on one chromosome that you cannot really construct genetic map. You cannot estimate their relative position towards the other genes, or they can actually sort independently. However, we know that this all A, B, C, and D are on the same chromosome, so we will consider that certain genes will be in different linkage group. So how do you proceed? How do you start? First, you look for those genes which shows recombination frequency less than 50%, and you have them outlined right here. So these are genes B, C, and gene D. So you see that these three genes shows recombination frequency less than 50%, so these three genes will form one linkage group. Now, you know that there is fourth gene, gene A, so let's compare recombination frequency of gene A with gene B, C, and D. If you compare recombination frequencies between A and B, you see it's 50%. A and C is also 50%. A and D is also 50%. What does it tell you? That gene A is far apart from all three. Gene A is far apart from all three B, C, and D, so gene A forms independent linkage group. Okay, so you can only construct genetic maps for B, C, and D. So if you start, to underline it, gene A is one linkage group, forms one linkage group, and genes B, C, and D form second linkage group. Now, how do you construct genetic map for linkage group 2? You proceed the same way as previously. You look first for two genes which are farther apart, which have highest frequency of recombination, and you see here it's gene C and D, which shows 28% recombination frequencies. So you know that gene C and D are farther apart, and therefore gene B will be in between of them. And if you look here, how do you know that gene B is between of them and it's not outside? Because you have distance between B and D and B and C, which is less than 28%. So it means that it has to be in between. And you see that gene B is closer to gene D. It's only 10% recombination than to gene C. So distance between B and D is 10 map units, and B and C is 20 map units, which gives you 30 map units. However, look here, your recombination frequency you observed was 28%, meaning it would give you 28 map units. So why it is 30? Why is the sum of distance between C and B and D does not give you different numbers than what you observe? You remember what we discussed before. Most probably there were double crossing over, which actually caused that observed recombination frequencies are lower than distance of these two, relative distance of these two genes on the chromosome. So let's now discuss three-point test cross. In three-point test cross, you can establish position of three genes on a chromosome in a single set of progeny, and you can also determine a double crossovers. You can detect them here, which provides you with much more accurate map distance. So based on this, you can immediately see that a three-point test cross is more efficient mapping test technique than two-point test cross. So let's look at this example here. How do you test it and what is the outcome? So if you have two homologs right here and you follow distance of gene A, B and C in one three-point test cross. Now we will again consider recombination, homologous recombination between two non-sister chromatids right here. And I will outline it on the side. So what can happen? You can have single crossing over between gene A and B. You can have single crossing over between genes B and C, and you can have double crossing over. And let's look at these two chromatids. So if you have on one chromatid, I will only outline those two chromatids, those ones which undergoes recombination. So one carries all dominant allele of genes A, B and C, and second carries all recessive alleles. So what would happen if you have single crossing over between genes A and B? So after the single crossing over between gene A and B, what would happen? You will get recombinant progeny which would have composition of alleles of this region A, B, C and A, B and C. What would happen if you have single crossing over between genes B and C? So you have single crossing over between genes and C right here. So in this case, you would have A, B and C, and second chromatid will carry A, B and C. So these are single crossing overs. Okay, so now what would happen if there is double crossing over? So we will have first and second crossing over. So we outline the chromosome after first crossing over. So after first crossing over, you will get A, B, C and A, B, C. Now, what if there is second crossing over? So what would happen? Now the crossing over will be between B and C, so you will obtain offspring which will have A, B and C, and here you will have A, B and C. And what you can see right here is that if you compare this double crossover to parental position, would you see immediately that basically the gene which is different from the parental composition is one which is in the middle. So recombinant chromosomes which result from the double crossover have only the middle gene altered. Do you remember when you did two-point test cross, double crossover brought two allele at parental position because you only have two alleles. However, if you have double crossover between three genes, if you follow three-point test cross, you can distinguish double crossover because the composition of allele of the three gene would resemble parental, with exceptions. The exception will be the middle gene. The middle gene will be different.